All right, so we are now being recorded, and I will begin my presentation. So in this fifth video teleconference for BIOL 105L, we will look at the schedule, the calendar, and then we will see what we've done so far. And finally, most of the time allotted for this video teleconference will be for review in a form of self-quiz for the current exercise that we are doing, the spectrophotometry of proteins or proteomics. Okay, introduction to proteomics. Right, sorry. So here's our schedule. We are now in February. We are doing our fifth video teleconference, so quite advanced as compared to the one in the calendar. Also, the exercise being done right now is Intro to Proteomics or Spectrophotometry of Proteins Exchange with Osmosis in the Plasma Membrane. So next week, we are going to have our last exercise before the midterms week. So I will be giving you your midterm project per group again next week in our video teleconference. So what have we done so far? How are we? We've done micropipettes. We're done with microscopy and then micrometers. Let me ask you here before we proceed. What are the important things to take note of when you do microscopy? First would be the visibility of your sample. How do you make sure that your sample is visible? When you're doing light microscopy. Anyone? From your exercise number 2 lab report, have you mentioned that one? How do you improve visibility?
Sir, ang sample ko dapat hindi sliced as thin as possible.
Yes, okay, so exactly. There's a difference between seeing the microscopic world versus the macro. No, the everyday things that we see them because they reflect light. Okay, and the light bounces back from them to our eyes. But when you are viewing something, a microscopic object under the microscope, we see it because light can pass through it. So you're correct, it has to be sliced thinly. No, otherwise, it will be very dark and you cannot see the structures of the cells in that specimen that you're viewing. Second, how do we improve contrast in microscopy? Contrast means that your specimen, whatever you're viewing, is very distinct from the field of view. So you can distinctly see it. So how do we achieve that? It's actually very common, and I know you do this one. You might just be thinking about it, so you better speak your mind now. You might be correct. What do we do so that the contrast will be improved? Or let me ask you, if you're the one looking at the cells under the microscope, what will you do so that the contrast will be good? You can distinguish it from the rest of the surroundings.
Sir, adjust the light, sir.
adjust the light so that, can you expound how?
Sir,
Increase, decrease?
um, it depends, sir, if unsa ang intensity nga kailangan, sir.
All right. So there's a point there, no? Adjust the light. I think some of you have also mentioned about dark field microscopy. The dark field microscopy is you can actually achieve that one with our microscopes in the laboratory, the compound light microscopes. You simply have to um, uh, adjust whatever is in the diaphragm of the stage. You can decrease the opening so that lesser amount of light will pass through the specimen. And I think you've experienced that when you use the microscope, when you lower the intensity of light, di ba, makikita mo parang the contrast increases even if you don't stain the specimen. Okay? Especially if you're not supposed to stain the specimen. Now, when you view pond water under the microscope, you want to see the moving cells. You don't want it to be very bright. Otherwise, it will be very difficult to distinguish them from the surroundings. You want it to be a bit dark. So you're correct. Adjust the light. And the second way, which I just mentioned, is if possible, stain your specimen. And that will usually kill the specimen, no? Because the stain is a chemical, no? that will kill the cells. Right. And of course, we know that we can adjust the color of light that we use. You use blue light to increase resolution, the resolving power, no? Or decrease the minimum resolvable distance. Or if you want to maximize the amount of light that enters the objective, you use the immersion oil in between the oil immersion objective and the specimens light. So we've also done using the hemocytometer. The checked versions of your lab report, I'm going to upload them after this. Okay, and later on you may browse through them because I've already checked them. Osmosis in the plasma membrane, and then right now we are going to review spectrophotometry of proteins. So we will have a self quiz again. So are you ready? In order to be ready, you might want your lab manual to be open, or to really test yourself. Don't. You might need a calculator because I'll ask you to calculate something later on. Calculator or cell phone. So are you ready, guys? Not yet ready. Again, this one is not going to be graded, okay? But it is for you to find out if you've missed something from the exercise. I think you will also get something from this because you are still in the progress of writing your lab reports. The deadline of which I've moved. Okay, so later I'll announce the new deadline for this exercise. So are you ready?
Yes, sir.
All
Yes,
right.
sir.
Yes, sir.
Go try,
Okay,
sir.
when I mentioned na move ang deadline, yeah, you answer. You answered. Because I feel maybe some of you are too pressured right now. No? Anyway, later you'll see the new deadline. So, question number 1 on spectrophotometry of proteins. By the way, I retitled it because Intro to Proteomics, although it studies about proteins, I think it's barely proteomics. If you know the definition of proteomics, okay, so it's barely that, but that's how the manual calls it. I say it's the spectrophotometry of proteins. Question number 1. Which samples had their protein concentrations analyzed?
Milk, sir.
What's milk? Okay. So milk, how many samples were there?
Two, sir.
All right. So not necessarily of different brands, but they could be of a different variant, no, of the milk produced by that brand. I hope you observe the nutrition facts label because later on in the exercise, it asks you to compare your findings on the concentration of protein of the sample versus what was advertised in the box. So I hope you did that one. Okay, so next. How do you call the assay that we used to analyze the concentration of the proteins?
Bradford assay, sir.
All right. So, can you say something very short, maybe one statement on what is a Bradford assay? Anyone?
Answer. Bradford assay is the method that determines how the solution contains protein. The darker the blue towards blue, that means there is a high concentration of protein in a certain solution. That's the purpose of the Bradford assay. Determining the concentration of the protein.
That is correct. No, so very important that you mentioned it has something to do with the color. No, a reagent will be added, and depending on the intensity of a color, a particular color, that's how much protein there will be. So it's a what we call it as a colorimetric assay. Colorimetric because it depends on how intense the color is. That's also the amount of protein that's present. So colorimetric assay. So that is correct. There's a reagent. So what is the reagent called? What did we mix with our samples to achieve that colorimetric result?
Answer. Bradford reagent.
Yes, so it's called the Bradford reagent because it's for Bradford assay. What is that Bradford reagent really? What's the name of
Sir,
that?
sir phosphate buffered saline, sir.
No, that is just for buffer though. No, that is just to dilute our sample. So it's not the phosphate buffer saline. Correct yung kanina, yung Bradford reagent. But what is it?
A dye, sir.
It's a dye. What's the name of the dye?
Kung ano, sir?
Sir,
Brilliant.
comma Yes,
Go
sir.
on.
Mr. Kato. Si,
Can you continue? What was that?
kaya na natin.
Kumase, what's the name?
Brilliant Blue G.
All right. So the Kumase Brilliant Blue dye is actually the Bradford reagent. Okay. So although it's called Kumase Brilliant Blue, in its form alone, it's not brilliant blue, right? It's not brilliant blue. But later on, it will turn brilliant blue if ever protein is present. Okay, so the name in itself is not the substance as it is. It needs to react first before it can be true to its name, which is brilliant blue. All right. So question number 4. Huh. Okay. How did the samples look like when there were proteins present? Pag merong proteins, what do we expect the color to be?
It changes to blue, sir.
Yes. Okay. So it changes to blue. Okay. So brought about by adding the Bradford reagent or the brilliant Kumase brilliant blue dye. Okay. How about when proteins were absent? What was the color? Or how did it look like?
Brownish, sir.
Yes, okay, so the Kumase Brilliant Blue by itself will appear reddish brown, no, or brownish. So you're correct. Before we go to that, what what is the reaction between the reagent and the protein? Why is it that it will turn blue? Can anyone tell us what that reaction is like, or how does it happen, the chemical reaction between the protein and the reagent? You don't need to elaborate on the chemical reaction, but just give us an overview. What is that reaction? It's in your manual. If you have your manual there, you try to read it in the introduction portion. What is the reaction like? Or what's happening? Why will it turn blue as you mentioned when there's protein?
Chelation or redox reaction, sir.
All right. So specifically, which parts of the protein react with the dye?
Ang side chains niya, sir. Ang R
All
group.
right. So yes, okay. The R groups or the side chains are the ones that are being attacked by the dye. So the reaction creates to our eyes a color of blue in the sample. Okay, so that gives the dye its name eventually. So what instrument? Because if you can remember, the first part is you were just asked to use your eyes and visually compare your sample with the other samples, no, with other standards. So that was just using your eyes, but we want to make sure. So what instrument was used to measure the concentration of the proteins?
One, sir. Spectrophotometer.
All right. So in a Bradford assay, you will need a spectrophotometer to really quantify the colors, no, because you cannot just rely on your eyes. So what if you're color blind, your blue looks like brown or red or the other way around? So we need an instrument, and that's the spectrophotometer in the case of Bradford assay. Okay, so now that we use the spectrophotometer, what value did we note? What was the reading that we note? It was a reading of what?
Absorbance, sir.
All right. The absorbance of the sample. Okay, so very vague, no? We'll arrive to what we're talking about here, really, no? So absorbance, or A. So which color and wavelength of light, because we are measuring absorbance in terms of light color. So which color or wavelength of light was set for the instrument to emit?
One, sir. 595 nanometers ang wavelength. Yellow line color.
Yes, that is correct. No? So a spectrophotometer, you can actually adjust it to what color, you just select the colors of the rainbow. And you need to know, of course, the wavelengths of those, because the spectrophotometer is not voice activated. You cannot tell it, oh, yellow. You have to know the wavelength of a yellow light, and then you input it. And then that's what the spectrophotometer will emit. So in our case, we used a 595 nanometer wavelength. so that the spectrophotometer will emit a yellow colored light. Why? Why yellow? Or why 595 nanometers?
Para ma-appear siya as blue, sir.
So that it will appear as blue? Why, why do you want it to appear as blue? Why not use blue light na lang? It will already appear blue without the sample. Why yellow? Remember, we are measuring, as you mentioned a while ago, absorbance. Sige. What if I use blue? How will it affect my reading? After all, the reagent will turn blue with the protein, right? So what if I'll just use blue, a shorter wavelength? Why must I use yellow? No answers yet? Okay, let me ask this in a different way. I have another question. If the color of your sample is blue, which color of light is reflected by your sample?
Blue, sir.
Blue. That's why its color is blue. So why not use blue? Why use yellow?
Kaya wala light mo absorb, sir, if blue ang gamitan.
Okay, if blue, baliktad ang result mo. Imbes na higher value for the blue sample, okay, magiging lower na yung absorbance if you use blue light. Okay, so sort of the opposite happens. So let's go back to question number 9. Why yellow? Why 595 nanometers?
Or naay light na ma-absorb sa sample sir, dili lang siya mo reflect bitaw.
Yes, because what's the color of your sample with protein?
Blue.
Blue. The peak absorbance of your sample with the protein, no? So you have the diprotein complex. The peak absorbance of that is 595 nanometers. That's why it appears blue, because it absorbs the yellow light and it reflects everything else na bluish. Okay? So I hope you get that one, no? Just like the logic of the color of leaves, diba? Leaves do not absorb green light. They reflect green light. Okay? They absorb everything else except for the green colors, for example. The same is true here. Your blue specimen absorbs all other colors except for blue. And most especially, it absorbs yellow. That's why we use yellow. Okay, so I hope you got that one. You have a question regarding that? So I hope you understand that in your writing, because you're still writing right now, you can incorporate things like that in your discussion, for example. Okay, so next, you remember there are protein standards. There are 7 of them. No, 1 to 7. What was their purpose?
Kuan sir, mura sila mga standard na kabalo naka pilay concentration para maka-construct og standard curve. Para na kay way of finding the concentration sa milk based on the absorbance.
That is correct. Nothing to add on that. Okay. Next, number 11. What was placed in the cuvette that we labeled blank?
PBS, sir.
All right. The buffer only, no? What else? There was a buffer, and then there was anyone? What's
Reagent sir.
the reagent? No, the Kumasi Brilliant Blue reagent. So in the blank cuvette, you have the buffer, you have the reagent, but you did not put any protein samples. Diba? Because that's our negative control. Diba? You're familiar with experiments. So in order to know if you're really successful, if the change is really because of that particular variable, you need to remove it from your control. Diba? So we remove the presence of protein in the blank. We only place the reagent and the buffer. Okay. So that's what's in the blank. And I think number 12, I already mentioned, the purpose is for negative control, okay? So that we would know what the color is in the absence of a protein. But there's still another reason. Can anyone mention it? If you can remember in the sequence of putting the cuvettes in the spectrophotometer, I started with a blank, di ba? And there's really a specific location there labeled B inside the spectrophotometer where you put the blank in. Why is it important to do that first before you put your samples?
Para makalibrate, sir? Or
Exactly. That's correct. So your blank is supposed to be zero absorbance. So you put it in your spectro, you set the 595 nanometer, you run it with a blank, no, you inside we don't see it, but the yellow light passes through the blank and everything passes through and the spectrophotometer will say 0A or zero absorbance to calibrate. All right. That's correct. Number 13, we diluted our samples, our milk samples with what?
PBS, sir.
All right, the buffer, no? Phosphate buffer saline. What was the dilution factor when we did this?
50 sir.
50. Now, 1 per volume, 1 is to 50. 1 for your sample and 50 overall. Okay. So you needed, for example, if you prepare 50 microliters, you need 1 microliter from your sample and 49 for the diluting agent for the buffer. That's correct. So what about this? Try to calculate. A sample was found out to have a protein concentration equal to protein standard 2. Okay. So what is its protein concentration in milligrams per milliliter? You might need to look at your manual what protein standard 2 is. Okay.
0.250, sir.
Okay, that's for protein standard 2. But the question is, what is the concentration of your sample in terms of protein milligrams per ml? Provided we diluted it similarly in the experiment.
12.50, sir.
All right, so that's the dilution factor, that's 50 times for the protein standard 2.250 milligrams per ml. So the answer is 12.5 milligrams per milliliter. All right, so that's the end of the questions for the quiz. I would like to continue by talking a bit more about the mechanism or the principle behind the spectrophotometer. So here we have the spectrophotometer. Let me use ink. Okay, so this is our spectrophotometer. Sorry. This is the spectrophotometer. And this is the chamber now opened for the samples. In the chamber, you can have, if I'm not mistaken, 5 samples and 1 blank. And then you have to cover it because it's very light sensitive. No, there is a detector inside that will measure how much light there is. Okay. And if you can also remember your electromagnetic spectrum by ito, okay, the color that we used had a 595 nanometer wavelength. Where is that? If you can see the values here that I'm underlining, 595 is around here. That's yellow colored light. Okay, so what we did is we turned on the spectrophotometer, inputted 595 nanometer, and look at this. The spectrophotometer is capable of emitting all colors of light. But once you set it to 595 nanometers, there is a sort of a grating that will only allow yellow light to pass. Okay, so now you see here in this illustration, only yellow light is able to pass because we set our absorbance value in the spectrophotometer for 595 nanometers. Okay, so for example, if you set your spectrophotometer for 750 nanometers, what color of light will pass through? Anyone? If you set the absorbance in the spectrophotometer at 750 nanometers, what color will pass through here?
Answer red.
Red. So you will have red light instead that will be passing through. And if you set your absorbance value in the spectrophotometer for 525 nanometers, what color of light will pass through?
Green, sir.
Green. All right. So that's the principle. Now we set first absorbance because that's what we want to use, to test how much concentration there is in our sample. And why did we choose yellow? Because we know that the higher the concentration of protein is in our sample, the bluer it gets. And we know that blue absorbs yellow. Okay, yellow to orange, somewhere there. but mostly yellow. So that's why we set 595 nanometers. Okay, so now that you are ready, you've now set the 595 nanometers. You may now put your blank. Okay, this is our blank. So if you notice, our blank is reddish brown in color because it only contains the dye, okay, the Kumase Brilliant Blue dye, and the phosphate buffer, and there are no proteins present. So when you put it and then you press B on the spectrophotometer so that it will test the blank cuvette, when the yellow light passes through, a majority of it will pass through, okay, the sample, and will be detected by the detector. Okay, the difference between the amount of incident light and transmitted light is actually an indication of the absorbance. So in this case, it looks like there's a bit of absorbance, but because this is the blank, you then tell your spectrophotometer, oh, this is 0. Diba? You then tell your spectro, this is 0. Because it cannot detect na 0 ba talaga or hindi. It's actually you thinking. You're just using this as a tool. So when you set it as 0, that's what we call calibration. Okay? You use your blank to calibrate your spectrophotometer. So may 0 ka na ngayon na basis. Diba? Now that you have calibrated, what's next?
Kuan, sir. Pwede na isulod ang protein standards.
All right. So now the ones with the protein standards, and then your sample, because you suspect protein in your sample. So if we put the sample in now, and then if this is the standard, let's use standard number 7, which is very blue. Okay. Look what happens. No light, yellow light is able to pass through. Okay. Meaning to say the detector doesn't detect any light. Meaning to say 100% absorbed. This translates to a very high absorbance value. So in your spectrophotometer, it will give you a number, 1 point something absorbance. Okay? So that's why this is very high absorbance value. No? So the spectrophotometer works that way. And I only have 2 examples here to show you the blank and standard number 7, so that we can sort of visualize how the spectrophotometer works. So are there any questions on this?
None, sir.
Okay, is that true for all? Okay. The spectrophotometer that we have is not limited to absorbance measurement though. Okay? It can also measure T. Nag-research kayo sa spectrophotometer, there's an A, there's a T. What is T? A is for absorbance. How much is absorbed? And what about T? If you came across that, yeah, you may mention it now. What's the opposite of absorbance? Like the one shown here in this blank, diba? It was able to pass through. What's the term for that? Sus, nandun na yung pangalan no, sa label. We have A, absorbance. What about T?
Kuan, sir, transmittance.
Transmittance. Okay, so the spectrophotometer may also measure how much light passes through. Okay, that's transmittance. Or it may measure how much light is blocked or absorbed. That's absorbance. And not only that, our spectrophotometer is also able to produce what we call as an absorption spectrum, okay, which in our next lesson in the lecture on photosynthesis, I will be talking about the absorption spectrum of the chlorophyll, and we can actually use the spectrophotometer to make just that, the absorption spectrum. So there are many uses of the spectrophotometer. Right. So with that, I'm done with my discussion. I hope that supplemented your exercise as you're writing right now. No? So if there are any questions on the spectrophotometer and this exercise, you may ask them now. But before that, exercise 5 is not anymore due this Saturday. What Saturday? Saturday is 6th. I moved the due date 2 days later. It's now due next week, Monday. Okay, so that's the due date for spectrophotometry of proteins. So it's your time to ask questions and clarifications. No questions, no clarification. So that means you understood the principle, no? Despite not coming here in school and doing it yourself, okay? But at least you know the steps, what should be done first. You calibrate it first. How do you prepare your sample in the cuvette? How do you clean the cuvette? Which side of the cuvette should you put where, right? Okay, that was shown in the video. So that's very important when you use the spectrophotometer. So wala talagang questions, clarifications?
None, sir.
All right.
Sir.
That's okay. So I think that's good. Okay, that means you understood. So the deadline is now moved, no, next week, Monday. So you may spend more time improving your narrative, especially in your discussion on this exercise. So our next exercise, though, will be about SDS page, no, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. If you've read your manual, that's our next exercise for next week. So with that, read ahead on that. And I thank you very much for joining in this video teleconference. I will post your results for exercise 3 after this, and you may view it in eLearn. So thank you very much, guys, and have a productive day ahead. You are now dismissed.
Thank you, sir.
Thank you,
Thank you,
sir.
sir. God bless.
Thank you, sir. God bless.